Right. Thank you, Aziz, for that very nice introduction, and welcome everybody, and thanks to the organisers for inviting me and giving me this opportunity to come and talk to you today about some of my interests in immunodeficiency research. It's a great thrill to be at this meeting. So, as Aziz said, I'm currently the chair of the IUIS committee on Inborn Errors of Immunity. So, I'm really going to talk about some of the advances that we've seen in understanding the genetics of inborn errors of immunity, or primary immunodeficiencies, and how these accomplishments have been made and how they're impacting patients at the diagnosis and the clinical management levels. So this is our committee. It's called, as I said, the Inborn Errors of Immunity Committee. It's overseen by the International Union of Immunological Societies, and it's currently comprised of many people on this screen and several people in the audience. This was taken in New York early this year when we had one of our meetings to discuss progress in the field. And it really is an international committee. As you can see from this map, where we've got well representation of members from North America, particularly in the US, in Latin America, across Europe, Aziz, as he indicated, from North Africa, colleagues in Israel, and also in Kuwait. And now, we've had a couple of members step down recently, so we've had a bit of a refresh in the committee. So we've welcomed these new members, two of whom are sitting on the stage on my right-hand side here, Isabel Metz, Anne Puel, Helen Sue, Mikko Seppänen, and Raz Somich. And we've also increased our reach globally with Mikko coming from Scandinavia and Isabel, of course, from Belgium. So it's a great collection of individuals. Of course, we don't cover all of the world, but we do our best to have representation in where we can and look to extend that globally as we move forward. So the objectives of our committee are several-fold, but one of the main responsibilities that we embrace is that we provide an up-to-date classification on all of the inborn errors or monogenic variants or mutations which underlie various immune dysregulatory conditions. These have traditionally been called primary immunodeficiencies, but we now appreciate that the clinical spectrum of these inborn errors is so broad. It's not just an immunodeficiency, it actually reflects immune dysregulation or immune dysfunction. So it includes conditions such as autoinflammatory conditions, autoimmune diseases, allergic disease, severe allergy, and even malignancy. So the committee really plays an important role in sort of curating all of the publications and, I guess, vetting to an extent the genes which we believe have been shown convincingly to be considered important clinically and perhaps even actionably from a diagnostic perspective. We also have several other objectives, and that really involves patient advocacy, education, liaising with various societies, but also with people like WHO, IPOPI, and so on to really push important issues forward when we can lend a voice and support to those movements. So I really don't need to introduce primary immunodeficiencies to this audience, but I'll just walk through some of the fundamentals to orient my talk. So immunodeficiencies or inborn errors are generally considered to be caused by single gene or monogenic mutations which affect the function of a single protein. And that is sufficient to result in disease. And that in itself, I find, is remarkable in the fact that our bodies are so complex and there's often redundancy within various biological systems. But in these cases, there is insufficient compensation or redundancy such that a defect in a single gene is enough to manifest clinically as susceptibility to infection, as autoimmune disease, as allergy. And these can be very severe and obviously often fatal. I guess traditionally, immunodeficiencies or inborn errors are considered to be very rare, and there's certainly studies which would support that. And this is a great paper from 2014 from Jennifer Puck and many people across the US, where they assessed the outcomes of the rollout of the newborn screening program in the US. This is where they measured the numbers of T cells by counting what are called the T cell receptor excision circles, or TRECs, in just over 3 million newborns. And you can see over here, or maybe it's easier to see here, that on average, the incidence of severe combined immunodeficiency as defined by T cell deficiency was about 1 in 60,000 births. So from that perspective, yes, inborn errors are very rare. But that's probably the extreme example, because we know that not all immunodeficiencies are detected by newborn screening, and that's shown nicely here. This is measuring your T cells on the x-axis. So yes, if you have SCID, of course you're going to fall in this quadrant here, but there's many other types of conditions in which you have normal T cell development but impaired function, and those cases are shown by these colours up here, and even some cases over here are not going to be picked up by newborn screening. So that 1 in 58,000 is a conservative estimate. And at the other end of the spectrum, we have this paper from earlier this year from Jean-Laurent Casanova's group, who actually found a particular variant in one gene called TIC2 explains tuberculosis in about 1% of Europeans. So clearly PIDs don't affect 1 in 100 people, but particular gene variants can. So approximately, I think a safe number would be 1 in 5 to 1 in 10,000 would be the incidence of inborn errors. But of course, there are those extremely rare individual cases of only one person or one family being identified to date with a particular variant in a particular gene. But the discovery and analysis of primary immunodeficiencies has been incredibly fruitful and incredibly informative to the fields of immunology, molecular medicine, therapeutics, because understanding the genetics of disease can have significant outcomes. It can obviously provide explanation for disease pathogenesis. It can enable diagnosis and therapy. From a research perspective, which is really where I come from, it enables analysis of the distinct functions of specific genes. And I think what's really important and perhaps underappreciated, it can lead to the development of really novel therapies, and not just for the patients from which those discoveries arose, but to other cases, other examples of more common conditions. So I'm just going to give you a couple of examples of that. I'm going to tell you about two boys. This strapping young lad was born in 1970 in the suburbs of Sydney, and grew up to be a aspiring surfer. Not so successful surfing, but he does enjoy life as an immunologist. He had a relatively normal childhood, did all those things that young boys do, went out, played games, lived a happy life, and continues to do so largely. This young man, on the other hand, was born a few months later in Texas and spent almost all of his life in a plastic bubble. And he, of course, is David Vedder, the boy in the bubble. And the fundamental difference between these two boys, apart from the hemispheres in which they were born, was that David Vedder had an error in a single gene called the IL-2RG, which encodes the common gamma chain. And as a result, David had X-linked severe combined immunodeficiency, and as such was kept in a sterile environment so that he wouldn't be exposed to any pathogen, so that he could live some type of life. So that was in the 70s. Unfortunately, David Vedder died in the very early 80s following complications from a bone marrow transplant. But that really brought to light X-linked SCID, and of course in the 90s, Warren Leonard's team at NIH identified the gene that was responsible for X-linked SCID. That of course wasn't known in David Vedder's lifetime. And following from that, Alain Fischer and colleagues in Paris developed approaches for gene therapy at that time, particularly for X-linked SCID, and these very impressive papers were published in the late 90s, 2000s. And of course, that has extended now the field of gene therapy, and I'm sure we'll hear talks about that over the next few days, to not just X-linked SCID, but several other types of immune deficiencies, which are life-threatening, often fatal, and can really only be cured by bone marrow transplant, or now gene therapy when appropriate. So that study of David Vedder enabled this sort of field to develop. But I also want to talk about perhaps what is considered the original primary immunodeficiency, which is X-linked Agammaglobulinemia. And this is the first report of this case, which was done by Colonel Ogden Bruton in 1952. And this was the study of a single boy who, in a very short period of time, had almost 20 very severe infections with pyogenic bacteria. He, interestingly, was not so prone to develop infections with viruses or fungi. It was mainly with bacteria. And keeping in mind at the times, there wasn't a lot of sophisticated technology to understand what was going on in this young boy. But what Bruton did discover was, by looking at serum electrophoretograms, the gamma globulin fraction, this bit here, what you see in healthy kids, was absent from this patient. So this was the first report of Agammaglobulinemia, and it's, as I said, often attributed as being the first description of a defect in host defense. What was remarkable at the time, though, in this paper, Bruton treated this young boy with essentially antibody replacement using serum from healthy donors. And you can see now, when you infused serum, you could detect some gamma globulins in this boy's blood. And obviously that is pretty much the breaking ground moment for the realization that you can use antibody replacement therapy for many types of immune deficiencies characterized by hypogammaglobulinemia. Again, not just X-linked agammaglobulinemia. So this study, I think in the 50s, also inspired an incredibly important discovery that came 10 years later in the 60s by Max Cooper, which was essentially the discovery of B cells. I get very excited about B cells. B cells is something that I'm passionate about and work on feverishly. So going back to 1965, lymphocyte subsets had not even been described at this point in time. But what Max Cooper had noted in this paper was that it was probably the case that there were distinct populations of immune cells that exhibit a division of labour in host defence. And he used this case of Bruton's type of X-linked recessive agammaglobulinemia to make this point, because he noted how there was a virtually complete failure of plasma cell formation and gamma globulin production, but immunological failure was only partial because these XLA boys had some features of intact immunity. And that actually led Max Cooper to elegantly show with Bob Good the existence of T cells and B cells 50 plus years ago. So that many years later, about 30 years later, led to the discovery of Bruton's tyrosine kinase, which is the gene that's defective in X-linked agammaglobulinemia. It was found to be key for signaling in B cells, so without that protein, you're unable to form B cells and, of course, unable to make antibody. And what that then led to was the development of drugs that specifically targeted BTK. These drugs are called ibrutinib, and they are currently used to treat B-cell malignancies, many different types of B-cell malignancy, whether it be plasma cell malignancies or leukemias or lymphoma. So a very effective drug. So it's quite remarkable to think that in the space of 50 or 60 years, from 1952, from the description of one patient, had led through to the generation of drugs that targeted that protein to treat malignancies. And I really like this quote in this review that says, what Dr. Bruton could not have known was that his discovery of XLA would open the door to a new target for B-cell malignancy therapy and provide the rationale for the development of the first ever BTK inhibitors to treat malignancies. So again, whilst being rare, the study of primary immunodeficiencies has done enormous good for molecular medicine. So that brings me back to the main focus of what I'm doing here, which is to introduce the committee a little bit more and what we do. So the Committee on Inborn Errors or Primary Immunodeficiencies has been around since the 70s, and it's seen many members, very revered leaders in the field, be members of that committee. And the committee meets every few years and provides an update, a publication update, informing the community of advances in essentially the genetics of inborn errors of immunity or primary immunodeficiencies. So you can see here over the last 15 or so years, there's been these publications every 2 to 3 years reporting these findings. And what's remarkable is the steady and then at times rapid increase in the discovery of genes which have been found to cause significant human disease in terms of immunological conditions. So going back to the early reports in the 80s, you can see it was fairly slow runnings, but then as we hit the 2000s and so, the number of genes being identified has increased rapidly. And we've just completed the next update, which has been hopefully being reviewed, and there's now 430 genes, plus or minus 1 or 2, no doubt, that we've missed, to now be well over 400 different types of conditions which can result in immune dysregulation. So this there's been this huge increase here in the discovery of genes, and that's come from the advent of next-generation sequencing, as well as as shown here, the rapidly declining cost to the consumer to have next generation sequencing applied. So 20 plus years ago, it cost an incredible amount of money to sequence the human genome. That, of course, was the Human Genome Project, which cost billions of dollars. And this is Moore's law. This is the rate at which of depreciation in computing world. The cost of genomics followed that for a little while, but then it just dropped off rapidly to the point where you can sort of get your genome sequenced for bargain basement prices, and no doubt that will continue. So the availability of massive, unbiased sequencing of your entire genome in the guise of whole genome sequencing, where you sequence every single base in your genome, or all of the exons, which are the protein coding regions, or you can just choose a panel of those 400 genes or genes of your choice to interrogate, has really enabled rapid gene discovery. But this has also revealed a few surprises, one of which I never really appreciated until you start getting into this field, is that it's very difficult to sift out what is a normal variant in your DNA, what might be shared by many other people in the population, and what really is a pathogenic variant. So this is a sort of typical figure you'll see in presentations, where if you just sequenced my DNA, much to my shock, I would discover that there's actually 5 million variants, any one of those which could be pathogenic, but hopefully that's not the case. But there's a tiered level of filtering through all that data to finally find the proverbial needle in the haystack and come up with at least a short list and eventually a defined variant which you can confidently call pathogenic. But it's not a straightforward process. It actually has opened up a lot of concerns about what we determine to be a pathogenic variant, how we can apply that to the clinic, and whether or not that can be useful in a management or a treatment perspective of patients. So to help this a little bit, experts, as shown in this paper here, 5 years ago published recommendations or guidelines by which should be followed, and this is really what the committee adheres to really ensure that the science that's behind these genetic discoveries is at a sufficient level or meets a particular criteria such that these conditions are met. So basically, the genetics has to be really strong, the biology or the biochemistry of the variant that you identify has to be very convincing, and there has to be good correlations between the gene defect and the functional defect with the clinical picture of the affected individuals. So with the access to genomics is great. It's certainly increased the rate of discovery, and you can see here, this is actually a figure I've modified from a paper of Isabel's a few years ago. Pretty much all of the genes that we are identifying these days, which result in immunodeficiencies, are being identified by next generation sequencing. That's what the red bar means here. So pretty much all the genes are coming out by that process. But we need to be very stringent to really filter out and be confident in what we're calling a pathogenic variant versus a benign variant, which may be present in more people than we realize. So what the committee does is classify these different inborn errors into various tables to simplify diagnosis and also understanding. So at the moment, as I said, we've just finished the latest report. There's actually 10 tables, which you can see here. I'm not going to go through them by table. Some of you would be familiar with these if you've read the previous updates. I'm just highlighting number 9 here because we've added this as a separate table now to be dedicated just to genes involved in bone marrow failure. So that's pulled some genes out of different tables as well as added new genes to that condition, to that category. And we also include phenocopies of inborn errors, which can actually occur due to somatic mutations. An example of that is autoimmune lymphoproliferative syndrome due to somatic mutations in the FAS gene, or autoantibodies. You can make antibodies to cytokines, and they can mimic some inborn errors as well. Okay, so accompanying these publications is this heroic classification table that my colleague Aziz publishes and curates essentially. Now, again, I'm not going through this, I'm putting this up here purely for convenience to show you that there is a classification publication which really enables simplification of the diagnostics and the complexities of those genes involved And also what Aziz has championed is the development of an app. Now, you should all have this app. It's actually fantastic. And it enables all of that can be done by the literature and from these publications, all on your iPhone or smartphone or something. So it enables that sort of work as well. So what's some other things I just want to touch upon before I finish are what I find some fascinating discoveries that have come from the genetics analysis of human immune deficiencies. One is that it's very easy to find a variant in a particular gene, but what's not so obvious is perhaps what that variant's actually doing. And I've just listed here 3 genes which can now been found to cause disease by different mechanisms. So for example, STAT1, and Anne might talk about this, was originally found to be pathogenic in a loss of function state, so both in autosomal dominant and autosomal recessive forms. It could cause Mendelian susceptibility to mycobacterial disease. That's what MSMD is. If you have loss of function in a recessive form, it also leaves people susceptible to viral disease. But then it was found to be also pathogenic if it encoded proteins with gain of function, so activating mutations, and it caused a completely different phenotype. And that's also true for CARD11 and this other gene, RAC2. So just to put that into context here, STAT1, if you have too little, you become very susceptible to mycobacterial and viral infections. If you have too much STAT1, you develop chronic mucocutaneous candidiasis and some autoimmunity. Likewise with CARD11, the first reports of CARD11 mutations were down this end of the picture. If you had recessive mutations, you had a combined immunodeficiency. And then a few years later, it was found that if you had a gain-of-function mutation, you actually had polyclonal B-cell expansion, very different to combined immunodeficiency. And very recently, there's these autosomal dominant hypomorphic mutations, which actually result in allergic disease. So the same gene can give rise to very different and quite complex clinical phenotypes due to different mechanisms and different patterns of inheritance. And again, these genes here, they can be pathogenic in different forms and different combinations and give either milder versions of the same disease if they're in a dominant form, or actually, in the case of PIK3CD or PIK301, quite different conditions as well. So there's this growing list of genes, and there's some more, which tell us you've really got to be careful and understand the consequences of these variants on the protein function so that you can confidently ascribe a condition to that genetic defect and know what you're really talking about. I have to wrap up, so I'm going to just tell you one other thing very, very quickly, is that next generation sequencing has really enabled gene-targeted therapies for inborn errors of immunity. So this is a whole list of drugs which are being used clinically for other conditions, so usually inflammatory conditions, arthritis, colitis, psoriasis, and so on. But they've been successfully used in a gene-targeted way for the treatment of different conditions. One is the inhibitors of JAK kinases, and another one is a blockade of CTLA-4, CTLA-4, no, abatacept is the drug I'm talking about, CTLA-4 Ig, which blocks activated T cells from being too pathogenic. And that was shown very recently, a new condition which could be treated by this. So I had a few other slides, but I'm going to skip over them and just jump to the end. And I'm happy to answer questions about those slides I skipped over. And just, again, acknowledge the committee. It's a great bunch of people. Welcome the new people who are coming along onto our committee. This is people I work with in my labs in Sydney, and all the various clinical colleagues and funders that we are fortunate to have funding from to do our genetic and genomic and immunological work in Sydney. Thank you very much.